he continue presenting with us. Thank you. Welcome, Mr. Davis.
Thank you very much. Thank you very much, Mr. Arthur, for that introduction. And I just want to take this opportunity to equally welcome our esteemed participants in this discussion today, whereby we'll be looking at package inserts. What diagnostic power does package inserts have, and appropriate utilization of package inserts. If well utilized, they have a very great impact in our diagnostic aspects as a lab or as service providers. Colleagues, apologies, just a bit as I get my screens to move, my slides to move. For those who are asking about yesterday's CPD points, they're still in the process. Just hold your horses. We are basically still within 24 hours. And it takes about 48 hours for all the CPD points or certificates to be availed to our participants. So don't worry. As long as your details were captured effectively as we insisted, it will be okay. Okay, thank you very much. As displayed on your screens, we are going to focus our discussion on harnessing the power of package insights in rapid diagnostic kits. Of course, package insights cut across. We have package insights in chemistry, package insights in hematology, but today's discussion just wanted to focus on rapid diagnostic kits for inference here and there so that we see how well we can have this discussion. As you have been informed, we equally have our LMSK dinner that is tomorrow, and you are all invited. Okay, thank you. On introduction, we usually point it out that accuracy in the lab actually begins long before a sample even reaches the testing bench, or even long before the first drop of a specimen is actually applied to that rapid diagnostic kit. Quality in the lab begins way before any of the aspects that I've mentioned above are actually put in effect. So one of the most essential yet often overlooked tools in ensuring quality, standardization, and defensible results is actually what we call the package inserts. And in most of my discussions henceforth, you'll see that, of course, I've just shortened package inserts to PI, so you'll see most of the PIs in the discussion. So don't get worried when you see PIs, it means package inserts for that discussion. So for any rapid diagnostic kit, the manufacturer or the supplier is obligated to provide a complete set of components that actually support correct test performance. And this complete set of components that are actually supposed to be provided by the manufacturer or any supplier include: one, the test device; number two, the buffer or reagents that actually accompany that test device; Number 3, it must have, of course, desiccants in that package, so that for the purposes of humidity control. Then number 4, it must have quality control materials, and this is where applicable, because not all kits are actually supplied with quality. In fact, most of the kits, rapid diagnostic kits, are not actually supplied with quality control materials. So where they are applicable, where they are available, they must be included in that supply chain as such. Of course, it must also come with clear storage instructions, and most of these storage instructions, you will either find them on the box or on the package inserts. And then finally, every rapid diagnostic kit must come with a package insert. So every rapid diagnostic kit box, every new box must come with a package insert. And that is where we are going to have our discussion centered on today. So package inserts contains validated instructions on specimen requirements, test procedures, interpretation criteria, performance characteristics, limitations, and quality control expectations. Of course, holistically, package inserts are a holistic description of every aspect of the kit performance. So in subsequent slides, we'll be also focusing on each of the individual aspects so that we see what it brings on board. So we say that consequently, when a rapid test kit is supplied without package insert, that represents a very fast diagnostic red flag. Your supplier gives you rapid diagnostic kits, and when you open those boxes, they do not have package inserts. That represents a very fast diagnostic red flag. Be very cautious with such like this. Of course, in the absence of package inserts, it might suggest compromised traceability, because yes, you are performing this test on what instructions or what is actually guiding the performance of this test. Also, it brings out potential non-compliance with the regulatory and quality standards, and also may have an increased risk of incorrect test execution. Okay, so without the package inserts, users may unknowingly deviate from validated procedures, misinterpret results, or apply kit outside its intended performance parameters. And these, in the long run, may lead to diagnostic errors that may directly affect patient management. So every kit supplied must, and we are using the word must intentionally because that is how it should be, must have an accompanying package insert, because that package insert is what will actually guide you on performance characteristics or performance parameters of that kit as such. So by contrast, the presence of the kit, the presence of the package insert and its proper utilization then anchors diagnostic accuracy. We have just focused on, discussed on what happens if the package insert is missing, okay? So this time we are looking at what happens if it's available and it's used properly. If it's available and is used properly, it anchors diagnostic accuracy. It ensures that the test is performed as validated by the manufacturer. If the manufacturer tells you, perform this test with ABCD instructions, the instructions have been there, the staff have been trained using those instructions, so performance of the kit must mimic the instructions that have been given by the manufacturer. Of course, in the long run, it also supports adherence to GCLP, data integrity principles. And finally, it also provides the scientific foundation required for correct test interpretation. How do you interpret your rapid diagnostic test? On what grounds do you base your interpretation on? So that is something that actually package inserts bring out very clearly. So in the long run, package inserts transform a simple rapid test kit into a reliable diagnostic tool, safeguarding both the patient safety and laboratory credibility. If you really have package inserts available and they're used properly, they're used as a foundation for training, They're used as a foundation for test performance. They'll actually ensure patient safety and laboratory credibility. We will look at that in the subsequent slides. So key information for conformity. As a person so much interested in quality outcomes in the lab, this is what I will actually focus on the most, because I look at all personnel involved in rapid diagnostic testing, including laboratory technologists and any other designated or default cadres. This was put here deliberately because not everybody who is actually undertaking the rapid diagnostic testing is actually a lab technologist or a lab professional, okay? So it means that whether you are a lab technologist or not, you must actually read, understand, formally acknowledge package inserts for every test kit that you are using. That is basic drive principle. And as I speak, I believe I'm speaking to laboratory technologists. If you work in environments where other cadres are actually allowed to conduct this rapid diagnostic kit, it means you are the testing point. You give guidance. Once you receive these kits, verify availability of package inserts, train these staffs using the package inserts instructions as given, then evaluate their performance based on the directives of the package insert. So this requirement actually applies to every new batch or lot, as manufacturers may update critical information such as performance characteristics, QC requirements, limitations, or procedural steps. Okay. This requirement is actually referring to the first statement that I've said up there. The requirement of you reading, understanding, and formally acknowledging package inserts. Okay. So the requirement is dictated by, it applies to both batch changes and lot changes. Why are we insisting? Whenever and whether it's a different batch but of the same lot, you must confirm that yes, the revision of that package insert in that new box actually conforms to what you have. If there are any updates, they are usually clearly indicated on the package insert. So we must always track these updates, as it's very essential in maintaining testing accuracy and ensures that staff do not rely on outdated instructions. Okay, so we must consistently make sure that we are monitoring updates on this package inserts so that we get any changes that might have been instituted by the manufacturer through the suppliers. Then we work on retraining our staff based on the updates that have actually been done as such. So all trainings must be conducted with direct reference to the package inserts, ensuring that staff learn and practice the validated manufacturer procedure without any deviation. You see, the kit, you can only ascertain kit performance if you are mimicking manufacturer specified performance aspects of that kit. If there is a procedure, mimic the procedure the way it is, because any deviation from that procedure can actually render your output invalid. Okay, so we must train all the users, all the consumers of these rapid testing kits with or using the package insert instructions. All the trainings must be centered around how to mimic the package insert recommendations. Of course, competency assessment in the long run must also be fully aligned with package insert requirements. Evaluating the staff members' ability to perform each step, interpret results, apply QC rules, and recognizing limitations exactly as outlined in the package insert. The package insert is a simple but most effective aspect that actually controls the performance of that kit as such. So when you conform to what we have just discussed up there, this actually then guarantees standardization of our practice. We do not want a situation whereby performance by one staff actually differs from performance of another staff. These package inserts, these instructions have been given by the manufacturers to ensure we are playing on the level ground, and that is what we are calling standardization of performance. Of course, it also goes a long way in reinforcing good clinical laboratory practice compliance, which cannot be taken lightly. Of course, it also goes a long way in reducing user-dependent errors. User-dependent errors are usually reduced when we have a platform of standardization, and we only have a platform of standardization in the form of package insert, which actually works as our standard operating procedure. Okay. And finally, it ensures that every individual performing rapid testing, RTK testing, is competent, accountable, and operating within the validated and traceable procedures. That is very important, and we need to actually conform to that. That was just finalizing on the introduction before I bring in the definition, important definition of package inserts. And we talk of package inserts are actually authoritative documents. These are authoritative documents because that is what you have been given by the manufacturer. Okay, so these are authoritative documents provided by manufacturers to guide the correct use, performance verification, limitation, storage, interpretation, and safety consideration of diagnostic kits. Okay, it's authoritative, as I have just highlighted, because it speaks what the manufacturer could have told you by word of mouth. Okay, it speaks to the performance characteristics of that kit. It speaks to the quality. It speaks to sample requirements. It speaks to all aspects of that testing kit. So it has to be available, and it has to be conformed to when it comes to matters quality. So these inserts are not optional for reading purposes. It's not optional, okay? These are core reference materials that support data accuracy, traceability, and regulatory compliance. They must be available. And if it's not all about availability, they must be read, they must be acknowledged, they must be put to use. Your testing procedures must be centered around package inserts. Even up to competency assessment when it comes to rapid diagnostic kits, it must be centered around that. So, having had an introductory bit of it, then let us then dive into the science. And we will be looking broadly at three categories. One, we'll be looking at the role of package insights in pre-analytical, analytical, and post-analytical phases of testing in rapid diagnostic kits. Then we'll be looking at the role of package insights in quality assurance in terms of rapid diagnostic kits. And we will finally be evaluating the performance characteristics of testing kits basing on the information that is on package insights. So, what is the role of package insights in a pre-analytical phase of testing? So, on my left, there's a snippet that I've just picked from one of the package insights, so that to help us or to guide us in interpretation of these matters. And of course, that is a Helicobacter H. pylori antigen test kit. That is Atlas Medical. Just for training purposes, we are not into other business. And it tells you that it gives you information with regard to samples preparation. And it also tells you or speaks to matters storage and stability. And these are very key areas that anybody interested in quality assurance of any given kit will always emphasize on. So in pre-analytical phase, we say that package inserts provide essential guidance that directly influences sample integrity and accuracy of downstream results. So we are saying that package inserts specify correct specimen type. As we can see from an example that we have it has given that samples, we need a stool sample, and it has told you that do not use watery or diarrheal samples. I'll be giving an explanation as to why. If a kit says that do not use watery or diarrheal sample, why does it have to state that? And just for clarity purposes, the kit can give you, for example, different aspects. Let us speak to, for example, HIV testing. A kit can tell you you can use serum, you can use plasma, or you can use whole blood. Okay? That is what the manufacturer has given. The manufacturer has given you all those options. But when it comes to verification of that kit, kindly note this: when it comes to verification of those kits, unless you are verifying your kit with all those array of samples, that is okay. You can utilize any. But if you are doing kit verification with serum, that is the sample that you'll be using in terms of testing, okay? If you decide that your kit verification, you are using plasma, then even subsequent patient sample testing, you'll be using plasma. If you decide that your kit is being verified with whole blood, then even subsequent kit patient testing will be done with whole blood. The sample type that you have settled on when it comes to verification, that is actually the sample type that you'll use when it comes to testing patient stuff. You cannot verify the kit performance using serum in the lab, then you test patients using whole blood, okay? Or you have used plasma when it comes to kit verification in your settings, then you are using whole blood when it comes to testing. So, colleagues, this is a very important point. The specimen type that you have used to verify that kit in your testing area, that is actually the specimen type that you will subsequently use to test your clients. Do not mix up that. So, package inserts also specify the minimum sample volume requirements, ensuring that adequate materials are reliable for reliable testing. Of course, it also speaks to the fact that proper specimen storage and stability, including acceptable temperature ranges and time limits before testing. I'll speak to the snippet that I put there. It has just clearly told you that storage and stability, store your kits at 2 to storage and stability, that is speaking to samples. It says between 2 to 8 degrees Celsius. We must make sure that we are mimicking that temperature, and the only way to mimic that is putting them in the fridge and monitoring the fridge performance. Of course, it has spoken to the fact that the reagents are stable until the expiry date stated on that. Down there, when it comes to sample preparation, the very last point, there is a point that samples can be stored at 2 to 8 degrees Celsius for a longer period of time, okay? Collect the stool sample in a clean container and use as soon as possible. If it's a collect once it's supplied, run the test as soon as possible. If you are not able to run that test as soon as possible, then kindly put those samples between 2 to 8 degrees Celsius. That is in the fridge. That is what the manufacturer has told you. So we must actually mimic what the manufacturer is telling you, okay? So it also speaks to the fact regarding handling requirements to prevent hemolysis, clotting, and contamination and degradation. That is very important. But on the fact that the manufacturer has said do not use watery or diarrheal samples, there's a slide towards the end of this presentation. I'll actually be bringing to you why it's that way. So it speaks to the fact that package inserts at this stage also ensures correct sample selection, preparation, and therefore fulfilling GCLP expectations for accurate sample identification, proper handling, and reliable pre-analytical data entry. We are just saying that as per the manufacturer instructions, let us try and mimic every aspect of that as it is. There's another snippet there that I'm just want to point out some very important aspects. On my right, on matters specimen collection, there is a statement: specimen collected may be stored for 3 days at 2 to 8 degrees Celsius if not tested within 6 hours. For long-term storage, specimens should be kept below -20 degrees Celsius. This one is very important, especially for, you see, getting how do we call it? Control materials for some kits like H. pylori is very difficult. Okay, so let us assume that on your testing plan today, you identified a positive H. pylori case. Okay, and for you to confirm that this is really a positive H. pylori case, you must actually send it for interlaboratory evaluation. If your partner lab tells you that this is positive, then you are confident that, yes, using your result and their result, you can confirm positivity of that case. So you want to store this sample, you want to store this positive H. pylori sample to use it as a positive control. You want to store it and use it as a positive control to evaluate kit performance. So what you need to do is, you cannot store this sample forever. The guidelines for storage of these samples must be as per the manufacturer instructions. It has just clearly told you that for long-term storage, specimens should be kept below. If you cannot achieve -20 degrees Celsius, then you cannot use that sample for long-term control matters, okay? Because it may degrade over time. But if the manufacturer has given clearly that with -20, you can have this as a long-term control, then you can use that as such. So we transition to analytical phase. Package inserts provide detailed validated instructions that ensure that the assay performs consistently and within manufacturer specification. That goes without saying. We must adhere to these requirements as it's essential for generating accurate, reproducible, and defensible results. So how then does it give guidance on that? We said, number one, that quality control requirements. Package inserts speak to the quality control requirements. Quality control requirements of those kits are actually found in the package inserts, and we must read those package inserts, clearly understand them for us to know the quality control aspects. Because there's a snippet on my left hand that I've just picked, and it speaks to internal control procedures are included in this kit. In this test, a green band appearing in the control region is internal procedural control. It confirms sufficient specimen volume and correct procedural technique. So the kit has given you that instruction. So it tells you that, yes, we have an internal mechanism of controlling or to act as a control to the performance of the kit. So it's your responsibility to ensure that, yes, you can identify that. If there's no control zone on your kit, then it raises questions. So in terms of quality control requirements, it tells you run positive and negative controls with each new kit lot, each new shipment, and as directed by the laboratory policies. Controls must fall within the manufacturer specified acceptable ranges. And where do you get these manufacturer specified acceptable ranges? It's on the package inserts. It also speaks to the fact of invalid control results require immediate repeat testing before any patient samples are actually analyzed. And how do we get this information? It's on the package inserts as such. Of course, through package inserts, they also give feedback on acceptable ranges of controls, just like what I've read previously. Package insert provides target control values and or acceptable limits. I've just mentioned an example in my previous slide. So results outside these ranges indicate proper assay performance. Do not report patient results until the issue is resolved and controls fall within limits. If you run a test and you find that there's no control line, how then do you go ahead and report patient results? You cannot report patient results until the control issues have actually been taken care of. Of course, number 3, package inserts speak to equipment and environmental requirements. Just to take your attention to the snippet that I have there that I picked from a COVID rapid antigen test, Luminostics. It says that the test has been validated for use in temperatures ranging from 15 degrees Celsius to 30 degrees Celsius. The test has not been validated for use in temperatures outside these conditions, and usage outside of this validated range may result in positive results, in false positive results or false negative results. Okay, this is very important information that you cannot miss, because if the manufacturer says that this kit must be utilized within 15 to 30 degrees Celsius, it means that is basically what we call room temperature. Okay, so we must then go further and ensure that our operations within the lab are actually within room temperature. Okay, how do we do that? We constantly monitor room temperature to make sure that it falls within what the manufacturer has specified as such. On that, it says that operate test devices or rapid kits within the PI specified temperatures and humidity ranges. Use recommended equipment only. There are things that appear on your package inserts that what is required but not supplied. It lists them, okay? All those things that have been listed there is actually what you are supposed to have at your end to actually produce the results as per specified aspect. Avoid testing in environments with excessive heat, cold, moisture, or dust. If the manufacturer has specified the given criteria, kindly make sure that we mimic that performance aspect as per the guidelines of the package inserts. Failure to maintain environmental conditions may lead to false positive, false negative, or invalid results. And that will be a situation of our own doing, because we will have necessitated that in one way or another, as such. So steps for resolving assay failures. If there's an assay failure, the package inserts actually gives us instructions on how we can resolve that. And it always surrounds between or repeat testing with a new test kit, verify sample quality and volume, recheck storage conditions of reagents, inspecting equipment functionality, reviewing QC performance and operator technique. And of course, all these corrective actions must be documented. If you do all this and you don't document, then it's more of meaningless. And I'll refer you to the snippet on my right hand that just says that this one I picked from FlowFlex COVID-19 antigen rapid test. It speaks to the fact that their package inserts include QC, environmental conditions, and invalid test criteria. For example, if a procedural control line does not develop in 15 minutes, the test is considered invalid and retesting with a new cassette is recommended. Of course, the manufacturer knows that if you do not conform to these aspects, or some issues might arise during your testing, they must be protected. And if they are protecting themselves, also protect yourself as a service provider as such. Of course, it speaks to matters procedural compliance. It gives the procedure, the testing procedure. Every testing kit, rapid testing kit, comes with its customized procedure. Do not assume that since I've been using H. pylori from company A, it must be the same. I'm a guru in H. pylori testing. No, it doesn't work that way. We must always refer to the package inserts, and we say that it's more of a caution. Let us not create our own protocols or shortcuts when it comes to rapid diagnosis or any other testing process. We must replicate these procedures as is where is, okay, without any change whatsoever. That is how we ensure that we have quality in rapid diagnostic kits as such. Of course, in post-analytical phase, we say that package inserts help standardize result validation. Of course, package inserts speak to the matter of validation criteria of results. It speaks to the matter of critical limits and reporting requirements, what is expected to us when it comes to reporting. Of course, it also speaks to the fact of documentation of limitations in the final report. And this is where the catch is, my colleagues. Every package insert comes with limitations of that test. And for you to be on the safe side, when it comes to matters rapid diagnostic kits. Every result that you give out to your patient, make sure that result actually clearly highlights the limitations of the test kit that you are using. And there is where, colleagues, we are having issues. Today, if you are taken to court that you are given out a false H. pylori test result, how do you defend yourself? You'll say, oh, this. Assuming that the testing procedure was adhered to 100%. The only way you can defend yourself then is on the matter of the test kit limitations. But the question will come then, when you issued these results to this patient, were the test limitations clearly highlighted? And were they clearly visible to the patient when you gave them out? Did the patient actually understand the limitations? Especially some of us who interpret these results to patients. When you interpret a result to a patient, don't be so quick to say, oh, you don't have H. pylori. No, okay? It might not be there at that time because the limit of detection has not been met. So, colleagues, we must always highlight, in fact, in bold, the limitations of every diagnostic on each of the reports that we give to our patients. So examples, result validation criteria from one of the kits that has been picked. It gives how do you then interpret a positive result, how do you interpret a negative result, and how do you interpret an invalid outcome. It gives you those instructions because you cannot know these instructions if you are not actually reading the package inside. These important features are actually only found on the package inserts. Of course, I can refer to this package insert snippet that I picked from somewhere. It speaks to the fact that the assay procedure and the assay result interpretation must be followed closely when testing the presence of H. pylori antigen in feces from individual subjects. Failure to follow the procedure may give inaccurate results. Number 2, H. pylori antigen rapid test cassette is limited to the qualitative detection of H. pylori antigen in feces. The intensity of the test band does not have linear correlation with the antigen titer in the specimen. This just speaks to the fact that, colleagues, let us not use qualitative outcome to give a quantitative explanation to our client. You find that, yes, we have a very dark band at the reaction point. You say, this is very high H. pylori. This is very high antigen titer. It does not work. There's no any direct linear correlation between qualitative aspect and the quantitative bit of the result. This is not a quantitative test. So interpretation is basically at the limit of a qualitative test. So it also speaks to the fact that a negative result for an individual subject indicates absence of detectable H. pylori antigen. However, a negative test result does not preclude the possibility of exposure to or infection with H. pylori. A negative result can occur if the quantity of the H. pylori antigen present in the specimen is below the detection limit of the assay, or the antigen that are detected are not present during the stage of the disease in which the sample was collected. The results obtained with these tests should only be interpreted in conjunction with other diagnostic procedures and clinical findings. Colleagues, step 3, 4, and 5, those steps alone can get you out of jail. And those steps alone can put you in jail. Why do we insist? For every diagnostic report that you give, kindly highlight the limitations of that testing kit. If you gave your report, for example, you gave your report and it has highlighted the limitations number 3, number 4, and number 5, and you go to test, and this client goes and tests somewhere else and it's positive, and you reported that is negative after some duration of time, this is the only thing that you can use to actually defend yourselves. We are in this process to make sure that we enlighten each other so that we know what is expected of us and what is actually required of us. So test limitations must be incorporated on analytical reports. This is the take-home point. Especially those of us who use laboratory information, it's hard if you are writing these things by hand, for those of us who are still doing manual. But if you have a laboratory information system, make sure that these statements, it does not necessarily mean that it's this. With respective kits, kindly tailor your statements so that it reflects the interpretation of that kit. Interpretation of a report should actually be done with limitations in the mindset. And we should continuously educate or enlighten our clinical team to actually make sure that they are aware of these test limitations when they are interpreting these kit as such. Of course, for commercial purposes, the moderator had indicated that tomorrow, 28th November 2025, we are having our LMSK dinner in Westlands, Pride Inn, Westlands, Nairobi. And it's always good to appreciate our sponsorship. It's not that easy to get a sponsor. And when somebody steps in to actually facilitate some of these aspects, it's good to appreciate it. And one of these sponsors is actually ChemLabs Limited. ChemLabs Limited have been seasoned or long-term distributors or suppliers in the country. Just visit their website, or you can visit their offices so that they have you have a look at what they are offering. And if it meets what you want, kindly just get in touch with them. Their contact information is as available down there. And one other such sponsor is Flowfarm Healthcare Limited, and it deals with the laboratory equipments and consumables. Those are some of their products as displayed on our screen, and it goes ahead to speak to the fact that they deal in instruments like microscopes, hematology analyzers, biochemistry analyzers, immunoassay machines, centrifuges, incubators, basically most of what we need for startup labs. Or if you want to upgrade, you must then look for one of these partners. Each of these partners have tailored solutions to your needs. It's just a matter of knowing what you want and how to get it. Of course, on the screen, we have contact information of the Nairobi, Eldoret, and Lodwar offices. Regardless of where you are just a phone call away. Of course, we have Keton Consulting. Keton Consulting, these are product sellers and also EQA and PT providers, solution providers. You can get them or have a look at what they offer in their website as displayed on a screen, or you can have a one-on-one discussion on the contract available on your screen as such. That is just part of what a few of the many products that they have in the market. In addition to EQ and PT plans, we have a bioassay 240 plus automatic biochemistry analyzer on my left hand. For those of us who are enthusiastic about histology, cytology, they have a spin tissue processor STP-120, and they have a modular tissue embedding center EC500. All these are solutions that are actually available at Keton Consulting. And of course, they have an STP-120. So where do you get this? Kindly just visit their website or through the contact information that is available so that you can have a further discussion with them on matters around that. Thank you. We continue with the science. So number 2, we look at the role of package inserts in quality assurance. And we say that package inserts are more than simple instruction sheets. They are validated manufacturer-provided documents that serve as the backbone of quality assurance in rapid diagnostic kits. PIs or package inserts play a critical role in ensuring that tests are performed accurately, consistently, and safely. Because at the end of the day, what runs in our head is patient safety. So we must actually conform to all these matters as such. So number one role of package insert is standardization of testing procedures. Package inserts give instructions on how testing should be done. That is your SOP. In most cases, you will not find any other SOP beyond what the manufacturer has given you. And I keep on insisting that yes, we must replicate what the manufacturer has given at all costs. So peers provide step-by-step instructions for test execution, specimen handling, and result interpretation. It also goes to ensures all operators, regardless of experience or cadre, perform tests uniformly. If the instructions are there, you must conform to those instructions as such. Of course, when we have a standardized form of instructions to conform to, it reduces operator-dependent variability, which is a major source of errors in RTKs. Most of the RTKs actually arise when we do not conform. If you are verified, if you have verified the performance of your kits and are up to date, up to standard, you are confident that with your verification report, you are confident with the performance of that kit. The other thing that then matters most is standardization of your testing operation. We should not have different outcomes when we have different operators of that kit actually performing the kit. We must have this standardized. Number two, it speaks to matters quality control compliance. Package inserts specify control requirements, acceptable ranges and frequency of QC runs. I've just, I had spoken to this. This is just a repetition for emphasis. It guides laboratories on lot-to-lot verification and internal QC testing. Ensures that the tests meet the manufacturer validated performance criteria before patient results are reported. We must really focus on that and have that in mind. Of course, it forms a basis. Package inserts form a basis for staff training and competency. I said that all trainings must be tailored to conform to what the package inserts actually says. So package inserts form the foundation for training programs and competence assessment. Staffs are trained to follow package insert instructions precisely, including specimen type, volume, timing, and environmental conditions. If colleagues, we cannot mimic what the manufacturer has insisted, and we cannot guarantee the performance of that kit. So of course, we go ahead that once we have trained our staff using the package inserts, we must also evaluate performance. We must also evaluate performance in terms of competence evaluation using those package inserts. We must use the package inserts to come up with appropriate KPIs to evaluate our staffs as such. Of course, fourthly, package inserts give instructions with regard to troubleshooting of failed outcomes and also corrective actions. Sawa. Package inserts provide guidance for resolving assay failures, including invalid controls, procedural errors, or unusual results. It goes further to help laboratories implement corrective actions systematically, preserving test integrity and ensuring patient safety. Every package insert, it gives you, in case the control line does not appear, what happens? Those are what we call instructions for troubleshooting and corrective actions. And how do we get that? We must then read our package inserts to have a proper understanding as such. Then finally, colleagues, we look at package inserts and kit performance criteria. The only way you will know the kit performance characteristics is by referring to the package inserts, okay? So package inserts serve as the primary reference for understanding the validated performance characteristics of every rapid kit that you come across, okay? They provide critical information that ensures the reliability, accuracy, and reproducibility of test results. So we must always make sure that we understand what these package inserts are talking about, and we apply those characteristics, okay? as that is the cornerstone in regard to quality assurance and GCLP compliance. So how then do they give the performance characteristics of kits? When you read your package inserts properly, you will find out that package inserts speak to the sensitivity and specificity of your kit. If colleagues, you come across a kit that the manufacturer has not given any details with regard to sensitivity and specificity of that kit, that is a red flag. Okay? So, package inserts report the clinical sensitivity. And when we talk of clinical sensitivity, we are looking at ability to correctly identify positive cases, and specificity, which is ability to correctly identify negative cases. Okay, so package inserts speak to clinical sensitivity and specificity. It must speak to that effect. If the kit does not have any information about sensitivity and specificity, then what was used to actually bring this kit to the market? It must have those concepts as such. So the above two matrices, which is sensitivity, clinical sensitivity, and clinical specificity, are usually determined during manufacturer validation studies and may include sample size, population, and reference standards. Okay, so what happens? It is your responsibility to make sure that during your verification process, you also evaluate sensitivity and specificity. So laboratory staff must reference these values to interpret results and context of clinical decision making. But of course, we must also verify. We do not just, if the manufacturer has told you that your kit specificity and sensitivity is above 90%, then it's also your responsibility to make sure that you verify that by repeating that process in the lab across the testing protocols as such. Of course, it also speaks, PIs also speak to limit of detection. Limit of detection. So the limit of detection specifies the lowest concentration of analyte that the test can reliably detect. Important for understanding why sometimes we come up with false negatives and determining whether the kit is suitable for low-level infection or early disease detection. This is very important. Even before you acquire a given kit, kindly look at published information regarding its limit of detection. Okay? Because if a kit has a high limit of detection, it means that there is a very high possibility of this kit giving you false negatives. Okay? Very important. And it's also good that using limit of detection, you can arrive whether the suitability of a kit suitability in terms of utilization in low infections or early disease detection. Okay? Very important, and we must follow. And where do you get this information? This information is actually on package inserts. You might also come to a point where you find out that the package inserts may provide limit of detection in units such as nanogram per mil or international units per mil or parasite density, especially for malaria kits. If the kit is actually speaking to the matters parasite or matters limit of detection, look at that very keenly, because especially for malaria MRDTs, look at what the kit has specified as its limit of detection. It's very possible for a kit to give a false negative result based solely on the limit of detection. So this goes ahead to explain why some stool or fecal tests prohibit utilization of diarrhea or watery stool. This is what I was speaking to. Why do some H. pylori kits tell you that do not utilize watery or diarrheals? It's always centered around limit of detection, because those diarrheal or watery stool, the antigenic components have been diluted. And when you dilute the antigenic components that you are looking for, they make it fall below the detection limit. Okay? So there is a very high possibility of getting a false negative result. That's number one. Number two, the manufacturer prohibits that because during the validation process, whatever was used was a fully formed stool. If the manufacturer used fully formed stool in terms of validation, it's also your responsibility to make sure that when you are doing your verification, you use fully formed stool. And when you are doing patient testing, you also use fully formed stool. That is how these aspects are actually done. So if the manufacturer specifies that you must use formed stool, then you must use formed stool. If there's diarrheal or water stool, then testing has to be suspended until otherwise, okay? Because you are actually in contravention of what the manufacturer has specified as such. Of course, PIs also speak to analytical measurement range. So it says it indicates the range of analyte concentration over which the test produces accurate results, okay? Beyond this range, results may be unrealistic or unreliable or invalid. Staff should ensure specimen selection and handling comply with package insights to stay within the validated range. That is something obvious that we need to confirm. Of course, testing kits also speak to the fact of cross-reactivity and interferences, okay? Because the manufacturer, post-market release, the manufacturer did extensive studies around the kit performance to the extent that they looked at what are the potential interferences that might come up with this. So PIs list potential cross-reacting substances, e.g. other infections, antibodies, or medications that may cause false positive or negatives. That is something that we need to fully understand before even we decide to use that kit. Of course, it also speaks to environmental or procedural interference, e.g. hemolysis, lipemia, may also be indicated to that. So understanding these limitations is critical for accurate interpretation and troubleshooting of your kit. You might be emphasizing on something, but at the back of your mind, you do not know that whatever you are doing actually is picked out by manufacturers as a limitation through the extensive studies that were done post-market introduction. So we must understand what can cause cross-reactivity, what can cause interference on the kit performance before even we put that kit performance, so that we shield the kit away from those interferences. Or we can also interpret our results conscious of the potential interferences that may arise. That is very critical, and we need to look at that as such. It speaks, PIs also speak to the fact of precision and reproducibility, reproducibility. Of course, when you talk of precision in RTKs, we are talking of repeatability and reproducibility. Repeatability is a matter of a controlled environment where one staff, same time, everything, but reproducibility, we are talking of intra-assay across board, okay? So PI may report intra-assay and inter-assay variability. Intra-assay is repeatability, reproducibility is inter-assay, indicating how consistent results are within a run or across different runs or operators, okay? This informs QC planning and helps staff recognize that when a test results may fall outside acceptable reproducibility limits. This is something that, of course, previously, for those of us who started with us, we had an extensive discussion about repeatability, reproducibility in RTKs and how we control that. So when it comes to verification, we must also verify the repeatability and reproducibility as stated by the manufacturer. If he has not stated that, then it's our responsibility through the confusion matrix. Once we have run our samples, we can actually come up with that. Of course, package inserts speak to stability and storage. Performance characteristics are valid only if storage and handling instructions are followed. Whatever instructions that have been given by the manufacturer, we must always insist that we replicate those in terms of daily performance. We look at it, deviations from temperature control, specified temperature controls, humidity, and expiration dates can compromise specificity, can compromise sensitivity, can compromise control line development. Those are some of the aspects that we must always look at so that we make sure that our performance for the kits is as good as possible. Then also, kits give lot-to-lot consistency. Package inserts speak towards lot-to-lot consistency. It gives instructions on how this was done and how you can actually replicate this in your testing point. Package inserts describe performance across different manufacturing lots, ensuring that QC verification is meaningful. Helps laboratories compare results between lots and track any deviations and anomalies. Colleagues, just for emphasis, we must read, understand, acknowledge, and apply instructions as evaluated on the package inserts. As usual, this is my parting shot for you colleagues, ladies and gentlemen. Package inserts are more than instructions. They are scientific backbone of every diagnostic assay. Each step, limitation, warning, and performance claim is grounded on rigorous validation by the manufacturer. When we follow these instructions as is where is and verify the manufacturer claims through internal QC, EQA, and lot-to-lot parallel lines, we uphold analytical integrity and protect patient safety. Harness the power of package insert as they guide standardization, enable reproducibility, and when coupled with proper verification, ensure that every result we release is accurate, defensible, and aligned with true diagnostic quality. Thank you very much, colleagues, ladies and gentlemen. To the moderator, I bring it back to you.
Thank you so much, Mr. Davis. This is wonderful. Such a strong discussion you brought. And so we want to go through our chat section. So many questions have been generated from this educative presentation. And so we want to go through all of these questions so that we are able to understand. Our first question goes to you, and it's from Shem Nyasende, and he is making a comment as well, and he's saying some good lessons. Let's use some specimen for verification and subsequent testing, not to use different specimen types for verification and subsequent testing of patient samples. Mr. Davis, are you able to hear me?
Yes, I can get you, Mr. Arthur.
Have you got that comment? Do you have something to say about that statement that Shem has given?
Yes. That is something that I had spoken to. And this goes straight for those tests that you find you have multiple, the manufacturer has given you multiple samples that you can use. For example, in HIV testing, you find that the manufacturer has told you, you can use serum, you can use whole blood, and you can use plasma, okay? So when these kits get to your lab, you are supposed to actually verify manufacturer claims, right? So if you are using, for example, serum to verify the manufacturer claims, you must utilize that serum when it comes to patient testing. You cannot verify your kit with serum and you use whole blood in terms of testing. So this is a take-home point to all of us that the sample that you have used, the sample type that you have used in verification That is the sample type that you will use in testing. Let us not interchange samples. If you have used plasma in your verification process, you must also utilize plasma when it comes to patient testing. If you have used serum in verification, you must use serum in terms of patient testing. Do not just assume that because we have been given 3 types of samples that we can use, you can pick any. No, whatever you have used in verification, that is what you should use in patient testing. Thank you.
Yeah, thank you. There's another question from let me check, from Catherine Linger, and the question goes, let's say that you have been requested to carry out a test on H. pylori, stool for ovarian cyst, and Salmonella antigen test, and the sample is stool. So my question is, which among these tests is considered to be done first? That's a question from Catherine Linger.
Thank you so much. I presume I thought she was going to add a culture test there, but basically without culture, any of these tests can be done fast. Because one, if you look at H. pylori and Salmonella antigen tests, these are tests that have different diluents, right? So you are scooping and using individual diluents. The only thing that you are using normal saline here is OC. So any of these tests can be done at the same time. But if culture was there, then culture should be done first. Or you should have scooped the sample for culture before you proceed to these other testing. But all these rapid diagnostic tests, they do not have a chronological criteria on how or why this should be done. As long as culture is not there, any of this can be done at first.
Thank you. Yeah, this is a common question, but you can give your response. You can educate us on the same. One question from Jacqueline Chepkemoi, and it goes, can MRDT be positive and microscopy negative at the same time? Back to you.
Thank you. MRDT negative, but microscopy positive. I've just spoken to that fact when it comes to limit of detection. Okay? Limit of detection, especially most of MRDTs usually specify limit of detection on the package insert. So you might find that, yes, this person is a true positive malaria case. So you will find it on microscopy, okay? But since the threshold, the low limit threshold has not been achieved, MRDT will give a negative result, okay? So this is something that we should actually look at as such. I think there's just a follow-up that is popping up in case that MRDT is positive and microscopy is negative. The gold standard for malaria testing is microscopy, okay? So MRDTs are actually most of them based on the proteinous components of the malaria parasite, okay? The shedding capabilities. So you might find that, yes, you have done testing. This case was positive before. They have been put on treatment. The blood side positivity has cleared. But since the shedding characteristics have not cleared from the system, you find that MRDT turns positive. It's very usual. These are things that people have experienced. Either way, it's just a matter of the shedding process. Whatever that was released into the bloodstream has not cleared, okay? So when you do microscopy, there's nothing. But when you do MRDT, because those protein components have not cleared from the system, you find that it turns positive. Back to you.
Thank you. Thank you. You're able to hear me?
Yes.
Thank you. There's another question from Nicholas Kipngetich, and the question goes, what steps should I take if a patient tests positive when I test in my facility, then she goes to another facility and it turns negative?
Thank you very much. I think I spoke to this when we were having our discussion, that colleagues, every test kit has its diagnostic limitations, and every result must be interpreted with those diagnostic limitations in mind. That's why I was saying diagnostic limitations of a given kit can either put you in jail or can put you out of jail. And it's as simple as that. Make sure that every report that you give, as long as your quality assurance processes have been conformed to fully, and you are so sure that this report actually represents the sample or the patient status that you have received, kindly make sure before you release those results, before you interpret those results to the patient, take the patient through the test limitations before that, okay? Because it's very possible you can do a test in the morning, probably the shedding characteristics or the threshold levels had not been achieved in your stool sample, you find that it's negative. This patient goes to another facility in the evening post a given duration of time, and you find out that they're testing positive. It's very possible. But then when you got that negative result, did you make that patient aware of the clinical limitations of your testing kit? Because this has to, especially if you are printing your results and you can incorporate those limitations on your, kindly go ahead and put those things. They must appear. Limitations of a kit must appear on your result, so that it's interpreted in the context of the sample as it is where is, okay? Because you cannot interpret the same way a result that was done in the morning and a result that was done in the evening in a different. That is a deviation of almost 12 hours. 12 hours are sufficient enough for something to transform or shed more aspects to achieve actually the low detection limit. So it's very possible. Just make sure, be confident in what you have. As long as your kits are verified, you can speak to the quality assurance processes that you have followed, you have replicated manufacturer performance, then interpret your result as it is where it is. Thank you.
Yeah, thank you. Let me go through other questions that are coming in, and we see there's a question from Edina Ombiro, and the question goes, is there any harm if I use the sample from SAT diluent direct to a slide for microscopy? That is over and cyst.
I believe even before Edna answered that, requested for an answer to that question, she already has an answer. Colleagues, let us just learn to do things as our SOP specified. If your stool microscopy SOP says it's normal saline, can't you use normal saline? Why then should you go? That's why we are saying stop creating shortcuts, mimic manufacturer instructions as they have been given, okay? So that is a general answer to you. I know most people do this, but to you, I'll say no, it's not advisable. Let us just conform to specifications as it's required. Thank you.
Yeah, thank you so much. Let me go through this chat again for another question. We have a comment from Jacqueline Chepkamoy. She says, nice presentations, nice presentation, questions well answered. Let me see if we have another question. Now, from Evans Otieno, there's a question that goes, is it wrong to verify 3 sample types given by the manufacturer just to ensure we can use any sample type where applicable? Are you able to get this question,
yes,
Mr. Davis?
Yes, I believe I know what he's asking. Of course, when it comes to verification of kits, it's possible you can verify them using different types of samples, but then verification is a cost, okay? Verification is a cost. So let us say maybe you are running 20 sets on serum. Then you are running 20 sets on plasma. You are running 20 on whole blood. This is a very significant cost. That is why most labs only identify one type of that sample and they stick with it. But it's very possible if you have the resources that allow, or you have so many kits that are lying there and utilized, it's very possible to do that. But verification is a cost, and that cost is so significant when it comes. That's why most of the labs only identify one kit, and mostly kits that give these options, you will find that most labs go for serum because it's easier. Then they just stick to that. If I verified my kit with serum, I'll stick with serum. As simple as that. No. So it's possible, it's very possible, but then there's cost implication on it.
Yeah, thank you. Calvin Okech is also giving his point of view on malaria diagnosis, and he says some diagnostic kits like MRDT can give false negative results for BS, for MPS. And when you do microscopy, you find malaria parasites. So as lab personnel, should we reject use of MRDT because they at a point mislead? And the question goes down, further, but generally that is the bottom line. You can comment on that.
That's why I pointed out that every kit has its own diagnostic limitations in terms of every MRDT should actually be able to specify its positivity starts at what density, because specificity, positivity or negativity of MRDT are actually pegged on the density of positivity in blood, okay? So if a kit has specified that, for example, malaria positivity density from 50 and above will be picked as positive, then even when it comes to verification of these kits, you must work out on this, because that is why when we are verifying MRDT, when you verify performance of MRDT, you don't do it like these other kits. You must be able to verify to the lowest diagnostic limit that has been specified by the manufacturer. So you'll find that you'll have some purely negative cases, confirmed negative cases as per microscopy. You introduce them this side. Then you find the very low positives on microscopy with very low parasitic density on this side. Then you find those ones with a very high parasitic density on this other side. So depending on the performance, you also evaluate. That is why for any kit, before it's put in use, kindly let us verify first. Some of these cases are arising because, one, we do not understand, or we do not know what the lowest diagnostic limit of the kit is. So you expect that every positive on microscopy should actually be positive on MRDT. It doesn't work that way. It can be positive on microscopy, but since the parastemia density is so low, it will turn negative on MRDT. So we must understand how to weigh the options. So the issue of saying that we reject, there's no rejection as long as you have verified the performance of your kits, you have trained the people utilizing these kits, and when you have a negative outcome, especially a negative outcome on MRDT, should actually be accompanied by test limitations speaking to that effect. Thank you.
Julius Ondieki is concerned with the standard Q because he believes it sometimes gives positive results for syphilis. Are you able to maybe comment on that?
It sometimes
gives false positive results for syphilis.
Okay, all right. Standard Q. Why is standard Q first utilized in maternity testing? Because it's very sensitive, but less specific. Okay, so it's, in fact, that kit is doing what is supposed to be there. That is the same way when we speak about TrinScreen as the first testing kit. It's very sensitive, so it picks anything and everything that mimics HIV. Okay, that is the work that is supposed to do. That is why every positive standard Q in MCH should actually be subjected to now specific confirmatory testing. Those kits, when you have very, 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 very high positives in terms of screening, that should not worry you. The kit is actually doing what it has been designed to do, to pick anything and everything that resembles syphilis, or to pick anything and everything that resembles HIV. So the subsequent now very specific tests are now used to rule out the other things. So you might find that yes, it has picking out very many positive, false positive cases in MCH for syphilis. But then you are supposed to subject that client to confirmatory testing. Those kits have been put there intentionally because of their sensitivity. They are just doing or picking out what they are supposed to pick in the first place. Because in terms of screening, you are not supposed to miss out anything that resembles whatever you are looking for. So they are just working as they are supposed to be. So any positive from screening should then be subjected to now confirmatory testing aspects. Thank you.
Thank you. Another question from Ivan Dire, and it goes, when selecting the kits for use in the lab, should the limitations of the different brands be a factor for consideration?
It is, and that's why I'm saying that for every lab, people have different standards of what they want. For example, you might say that you as lab A, you want a kit that has specificity, sensitivity of 90%, specificity of 95%, limit detection level of around this one. Those are your standards. So when you go to the market or when you put out a tender, you make those requirements very clear so that whatever suppliers that are coming on board should have their kits actually conforming or above whatever standards that you have set. So that one is organizational based. It's you to define what you want. If you want any kit, for example, at 50-50 for sensitivity, it's up to you. It's you want. But then before you decide on anything, put the patient's safety first in mind. The kits that should be brought in should actually have the highest sensitivity to pick out anything or everything that mimics whatever you are looking for, and should be very highly specific, okay? Should be very highly specific to what you want, high specificity, and should have the lowest diagnostic limit so that everything that is positive, the kit should have the possibility of picking it out. Thank you.
Yes, thank you. Another question from Vincent Ochieng, and the question is, why is TrinScreen HIV testing kit having high false positive reactivity than Determine? And why was Determine kit phased out? Maybe you can give your comment on that.
On why Determine kit was phased out, those are politics. Those are regional politics. I won't speak to that. But on why TrinScreen is giving high positive, false positive results. Colleagues, I've just spoken to this and I've said the very first kit in HIV testing is a screening kit. And a screening kit should have the highest sensitivity to the sense that it picks out anything and everything that looks like HIV, okay? Every component in your system that behaves like or looks like HIV should actually be picked by the very first screening kit, okay? That is why there are 3 tiers of testing. The second tier and the third tier, those are very specific, highly specific kits. Okay? So whatever has been picked as positive, for example, with the TrinScreen, which is a very highly sensitive kit, picks everything and anything that looks like HIV, should actually be confirmed by the second and the third kit. Okay? So when TrinScreen picks very high false positives, that is actually the work that kit has been put there to do, to pick anything and everything that looks positive. so that that positivity will then either be qualified or disqualified by the presiding kits. Colleagues, let us get this understanding. This is HIV screening with three tires is more of an RRI. You are screening masses. So if you are screening masses, you must have a kit that is very sensitive and it picks everything and anything that resembles HIV. That is the intention, okay? So that it's just put there to make sure that we do not miss any potential positive aspect. But then once the positivity has been established with the screening aspect, it then has to be confirmed by the subsequent. So the issue of it there being so many false positive in terms of screen, it shouldn't worry you as a service provider. That is the very reason as to why, the very basic reason as to why that kit is there, to pick anything and everything, whether HIV or not, as long as it behaves or resembles the HIV performance characteristics. Thank you.
Thank you so much, Mr. Davis. It was a very educative session for us, the medical laboratory fraternity. And we also thank our participants for being patient up to this moment that we are concluding our presentation. And Mr. Davis, allow me to bring you back to give us your parting shot before I adjourn this session. Welcome back.
Thank you very much, Mr. Arthur and LMSK fraternity, for this opportunity, of course, to discuss package inserts. Package inserts, colleagues, ladies and gentlemen, are very core to the operations of our labs or to your performance as a laboratory technologist. Let us not make assumptions. These are very basic things that we really need to have at the back of our hands or the back of our minds so that we can actually translate. The take-home point from this discussion is, let us understand the demands of the package inserts. Let us understand the demands of the manufacturer. Let us replicate the manufacturer demands in our testing points. And if we fully replicate these demands of the manufacturer in our testing points, of course, we will have attained the expected performance, and you will confidently speak to the reliability and the quality of result as a testing individual or as a testing lab. Okay, kindly colleagues, let us conform to that. Let us not develop shortcuts. Let us not create our own procedures. Let us conform to what the manufacturer has said, as is, where is. That is the take-home point. Thank you very much.
Thank you so much to our presenter, Mr. Davis. And we also spotlight our partners whom have made it possible for us to have the Friday dinner, and that is the Chem Labs Limited, Keton Consulting, Intermedica, and Flow Farm Healthcare Limited. Till next time, we will inform you when we will be having another webinar presentation. Otherwise, we hope to meet you all tomorrow in our dinner gala at Westlands Pride Inn in Nairobi. And therefore, anyone who still is planning to attend, kindly register and make sure you come early. We'll be starting at exactly 4 p.m. and therefore we expect you to be there by 4 p.m. See you there, and thank you. Have a good evening.